Hello, everyone. Welcome to today's live broadcast, Therapeutic Protein Separations using Diamond-Based Wide-Pore HPLC Columns. I'm Laura Bush, the Editorial Director of LCGC, and I'll be your moderator for today's event. We're delighted to bring you this web seminar presented by LCGC and sponsored by Diamond Analytics. Diamond Analytics expands the existing range of analytical capabilities in separation science by providing diamond-based solutions that allow for the exploration of novel chemistries. As a result of breakthroughs in diamond technology, Diamond Analytics high-performance liquid chromatography columns offer expanded pH range capability from pH 1 to 13, elevated temperature ranges to enhance protein recovery, increase longevity, and novel selectivity without compromising efficiency. Specifically, Diamond Analytics columns can be run in extreme conditions of pH and temperature. With longevity in mind, these columns can be regenerated for repeat use, leading to a longer, more productive life and increased total cost savings. We have a few housekeeping announcements before we get started. The webcast is designed to be interactive, so we encourage you to ask questions. You can submit questions at any time by typing them in the Q&A box, and you can find that by clicking on the red Q&A widget at the bottom of your window. You can enlarge the slide window by clicking on the small green icon in the upper right-hand corner, or by hovering your mouse over the lower right-hand corner and dragging the window to the desired size. The slides will advance automatically during the web seminar. And if you have any technical problems viewing or hearing the presentation, please click on the question mark Help widget in the dock at the bottom of your window. We now have a brief polling question for all of you in the audience. You may click directly on the screen to submit your answer. Our first question then today is, how many samples do you analyze weekly? Fewer than 10? From 10 to 100, or more than 100. So once again, you can click directly on your screen to submit your answers. How many samples do you analyze weekly? Fewer than 10, from 10 to 100, or more than 100. All right, let's take a look at those answers. It's kind of evenly split over the various options there. Looks like most of you, though, either do fewer than 10 or from somewhere from 10 to 100. Okay, very good. I would now like to introduce today's speaker. We are very pleased to be joined today by Dr. David Jensen. David Jensen currently serves as a lead research and development scientist at Diamond Analytics. Dr. Jensen received his PhD in analytical chemistry from Brigham Young University. He has presented at multiple industry events, including PitCon and HPLC. His work on diamond-based chromatography columns and other areas of scientific research has also been published in 23 peer-reviewed publications, including the Journal of Chromatography and Analytical Chemistry. His doctoral studies were focused on surface modification and coatings of carbonaceous materials, including diamond, graphite, and carbon nanotubes. He has completed work on a number of patents in the following areas: modified diamond particles, surfaces and methods, and thin layer chromatography, plates, and related methods. David, thank you for joining us today. You have the floor.
Thank you, Laura, for the introduction. Also, thanks to the viewers for being here to listen to the discussion on our new Flare Wide-Pore diamond core shell material used in the analysis of therapeutic proteins. This work was done in collaboration with Zsombor Fekete and David Guillaume of the University of Geneva. As stated, I'm Dave Jensen, and I'm pleased to present this information. Diamond Analytics, a U.S. synthetic company, designs, develops, and produces unique HPLC flare wide-pore columns that are not based on silica. We expand the existing range of analytical capabilities in separation science by providing unique diamond-based solutions that allow for the exploration of novel chemistries. Our parent company was founded in 1978 and is currently the leader in diamond cutting tools for oil exploration. Diamond Analytics, a division of U.S. Synthetic, was founded in 2006 when we saw an opportunity to use our diamond knowledge in the realm of liquid chromatography. As such, we produce diamond-based HPLC columns. So why diamond? When people think of diamonds, they think of that pretty rock on a lady's finger. Unfortunately, but fortunately due to costs, we're not using gem quality diamonds in this type of application. Diamond is known for its hardness, thermal and chemical stability, and thermal conductivity. Here's a physical comparison table of diamond against some frequently used chromatographic materials. It can be seen that from the table that the density is similar to alumina, the thermal expansion of the material is small, its thermal conductivity is enormous, and it is stable to thermal degradation. Here is a basic scheme of how the particles are built. The core is made out of a carbon sphere to which a uniform layer of polyallylamine is deposited. Following the deposition of PAM, or polyallylamine, a uniform layer of nanodiamond is placed onto the surface. This process is termed layer by layer, and it's done until the particle is grown to its specified diameter. Lastly, the now grown nanodiamond containing particle is functionalized with C18 ligands to make it suitable for reversed phase liquid chromatography. Here are the physical dimensions of the flare particle. In this slide, it can be noted that there are 4 different types of flare columns. This presentation will focus on the flare wide-pore material. From the diagram, it can be noted that the carbon core is 3.4 microns in diameter, onto which a 0.1 micron diamond shell is placed upon, thus producing a final particle size of 3.6 microns. The average pore size of the finished flare wide-pore material is 250 angstroms, which is suitable for large molecules, such as biomolecules, as will be shown here in this presentation. Lastly, the usable pH range of this material is 1 to 13, with a temperature limit of 100 degrees Celsius. Even though the flare material has C18 ligands attached to the surface, the inclusion of a polar amine at the base of the C18 moieties impart unique selectivity, thus the term mixed mode. The red point in the hydrophobic subtraction model, or HSM plot, is the flare material. It stands out of the main grouping, which is composed of about 600 other types of C18 columns. It should also be noted that due to its unique selectivity, traditional column screening procedures may not allow the chromatographer to see the full benefit of using a mixed mode material. As stated earlier, the usable pH range is 1 to 13. Since most protein separations are done at low pH, using TFA as a mobile phase modifier, a stability graph was plotted. Human apotransferrin was used as the target protein in this study. The separation was run at 60 degrees Celsius using a mobile phase that contained 2.0% TFA, which produced a final aqueous pH of 0.7. It can be noted that close to 10,000 column volumes were ran through the column without any major reduction in the retention factor of apotransferrin as demonstrated in the plot. So to recap, here are some of the key data points of flare. As shown, the flare has unique selectivity and stands alone in the HSM plot. We show that the column is stable at low pH by running a large amount of pH 0.7 mobile phase through the column. It's also stable up to 100 degrees Celsius. It is reproducible batch to batch and column to column. It's LCMS compatible. It's also compatible with 100% aqueous to 100% organic mobile phases. It's produced and manufactured in-house using lean manufacturing methods through a proprietary layer-by-layer process as discussed, and is synthesized with 99.999% pure sp3 nanodiamonds. All columns can be operated on standard HPLC systems and also can be used on UHPLC systems. So most chromatographers are aware of current trends in column technology. Over the past 15 years or so, the particle size has decreased. Accordingly, this decrease in particle size has shown an improvement in column efficiency and has allowed for an increase in sample throughput. With smaller particles, higher flow rates are attainable without a substantial decrease in efficiency. However, at times, the high flow rates create high column back pressures. To overcome these issues of higher column back pressures but maintaining efficiency, core shell technology was developed. So, as shown in the schematic at the beginning of the presentation, flare particles are constructed to produce a core shell material. I believe that most of the viewers of this presentation are interested in how this material is useful for their specific biomolecules of interest. Since a large class of biomolecules have a large molecular weight, it can be inferred that the size of the molecule will not interact appropriately with traditional small pore HPLC columns. The flare wide pore material, with its average pore size of 250 angstroms, is designed for biomolecule separations. Additionally, with the review of how the particle is constructed, it should be noted that there are no silyl groups because the material is not based upon silica. With this the retention mechanism of flare wide pore material is based upon both hydrophobic and anion exchange mechanisms. To further the benefit of using this material for your separation purposes, the material is stable at high operating temperatures and offers the known benefits of core shell particle technology. Core shell particles have beneficial properties when compared to totally porous particles. The ability of the analyte to diffuse rapidly in and out of the particle allows for improved separation potential at higher flow rates. Because core shell particles have a tight particle size distribution, they are able to be packed better, which allows for a lower A term in the Van Deemter equation. Two downsides, though, to core shell particles is they show reduced retention and loading capacity, which is an artifact from the reduced surface area because it contains a solid core. Okay, we have a few graphs here. They demonstrate how loading capacity and efficiency relate to the size of the shell on the core shell material. Because the flare wide pore material is composed of a porous diamond layer that is placed upon a carbon core, the loading capacity of the particle is reduced when compared to a fully porous particle. As shown in the left side plot, as the core shell to shell ratio increases, the loading capacity decreases. This is due to a reduction in the available surface area for chromatography. Another interesting fact is that as the shell is reduced, the efficiency increases, which here is measured in plate height. So as the shell becomes thinner, the efficiency becomes greater. Now just a little more theory. As stated in the last slide, as the shell becomes thinner and thinner, the C-term contribution in the Van Deemter curve becomes flatter. This is important because if one wants to increase sample throughput, one lever that can be used is to increase the flow rate. As can be seen in the Van Deemter plots, as the shell thickness is reduced, so is the C-term. This allows for efficient separations at higher flow rates. One more important aspect of the Van Dienter curve and how it relates to the size of the molecule of interest is demonstrated here. For protein separation, the C term is the most important contribution to fast and efficient separations. It is known that larger molecules diffuse slower than smaller molecules. As such, the use of core shell material is advantageous because of the reduced amount of material the analyte has to diffuse through upon entering and exiting the particle. In the past few slides, we have discussed the typical Van Dienter theory and its application in determining the optimal flow rate for the target efficiency needed. However, unlike some small molecule applications, proteins are not usually separated under isocratic conditions. As such, the use of a Van Dienter curve is not applicable. In order to understand the importance of gradient profile time and its effect on the separation, we will use the metric of peak capacity as a means to measure efficiency. As noted in the first equation, peak capacity is based upon the resolution of 1 between consecutive peaks and their widths at half height. So now back to Laura for a polling question.
Great. So yes, we have another polling question for all of you in the audience. Once again, you can click directly on your screen to answer. So the polling question says, what temperature do you generally use in your separations? Lower than 35 degrees Celsius, between 35 and 60 degrees Celsius, or higher than 60 degrees? So again, what temperature do you generally use in your separations? Below 35, between 35 and 60 degrees, or above 60 degrees. All right, so let's pull up those answers. All right, so the vast majority of you are using temperature between 35 and 60. David, did you want to comment on that?
Yeah, that's what we were expecting. In the next few slides, we're going to be looking at methods development regarding proteins and trying to separate them from different types of variants. And in our studies, we saw that 35 to 60 degrees was important. However, we do have some applications where temperatures above 60 degrees are beneficial.
Okay.
Okay, there is a lot of information in this slide, as can be seen with the 4 different plots. We are going to go through and tell a story of how one would go about developing a method using some model proteins. Also, to use the information that we learned on the previous slide regarding peak capacity, we are going to look at the effect on peak capacity as a function of gradient time and temperature using cytochrome C, human transferrin, and bovine serum albumin as the model proteins. Temperature is an important parameter to change while trying to develop a method for a protein separation. Firstly, elevated temperature can decrease secondary interactions that may occur at lower temperatures. Additionally, temperature improves analyte diffusion, which allows for faster transport of the analyte in and out of the particle during chromatography, resulting in an improved separation. Another important parameter is the addition of an ion pairing agent, such as TFA, to the mobile phase. This is important because it increases the efficiency of the protein separation by improving the hydrophobicity of the protein via ion pairing with positive moieties on the protein. To emulate real-life separations, the column was thermostated at 30 and 50 degrees Celsius. 0.1 to 0.5% TFA was added into the mobile phase, and the gradient profile time was studied between 4 and 44 minutes. These chosen parameters are fairly common in developing a protein method. The aim of this experiment was to understand the influence of gradient steepness on peak capacity as a function of TFA concentration and temperature. First, the impact of temperature was investigated as shown in the first set of chromatograms. Several gradient spans were studied, and peak capacity was determined at 30 and 50 degrees Celsius when applying 0.2 milliliters per minute flow rate and 0.2% TFA. The first take-home message from this experiment deals with the influence of temperature. It is demonstrated that peak capacity does not change with studied temperatures. This is an interesting aspect because usually peak capacity increases respectively with an increase in temperature due to the reduction of ion exchange interactions that can occur on silica-based materials. The data suggests that ionic interactions are probably negligible under the study conditions. The benefit when working at lower temperature is the reduced risk of thermal on-column degradation of larger proteins. Another important aspect of the study is peak capacity reaches its maximum when the gradient profile time is around 15 to 20 minutes. This suggests that longer gradient times are not needed. Lastly, there is no noted decrease in efficiency when changing the flow rate from 0.2 milliliters to 0.4 milliliters, as shown in the lower right-hand peak capacity plot. As such, sample throughput can be increased without losing resolution and efficiency in your separation when working at higher flow rates. In the last slide the TFA concentration was set at 0.2%. Here, the influence of varying TFA concentration was studied. Traditionally, TFA concentrations of 0.05 to 0.1% are used when working with silica-based materials. TFA is used to limit the ionic interactions that are seen between residual cyanol groups and positively charged groups on proteins. Furthermore, TFA concentrations up to 0.5% can be used to help solubilizing large or hydrophobic proteins. As shown in the flare Wide-Pore column peak capacity plot in the upper right-hand corner, a drastic improvement is noted when the TFA concentration is increased. In this study, at least 0.2% TFA in the mobile phase is needed to exploit the full benefits of the flare Wide-Pore column. Additionally, an increase in TFA causes an increase in retention of all the modeled proteins. This is more than likely due to two effects. Firstly, an overall increase in hydrophobicity of the protein occurs upon ion pairing of TFA with the protein. Secondly, the surface of flare Wide-Pore under acidic conditions is positive. This allows for ion pairing to occur between the particle surface and TFA, resulting in an increase in hydrophobicity. Here is another study that looked at the use of various concentrations of TFA and the influence of such. The gradient profile time and column temperature were the same for all of the chromatographic runs. It should be noted that at low TFA concentrations, that is 0.025 to 0.05, from 0.025 to 0.05% TFA, the resolving power of the column is reduced. However, at TFA concentrations above 0.1%, the ability to produce sharp chromatographic peaks is evident. It should also be noted that as the TFA concentration is increased, the overall retention of the presented histones increases. That is, when comparing 0.1% TFA to 0.5% TFA, there is about a 25% increase in retention. This is possibly due to the items that were discussed on the previous slides regarding ion pairing of TFA with either the protein or the surface of the particle. Accordingly, as the data suggests that as one is developing a method, the concentration of TFA should be varied in order to produce the desired separation. To further support the core shell idea and the ability to improve sample throughput, here we demonstrate that at a traditional flow rate of 0.2 milliliters per minute for a 2.1 millimeter ID column produces nearly the same chromatography when flowing at 1.0 milliliters per minute. The target analyte for the study was insulin. The top plot is an overlay of both flow rates that have been adjusted to give relative retentions. As such, one can note that in the comparison, and when comparing the chromatograms, they are nearly the same. The lower left-hand corner plot demonstrates that a 5-fold increase in sample throughput is realized without a significant reduction in efficiency. So to rehash what was just presented regarding methods development optimization when comparing to conventional silica-based materials. First, high temperature separations are not needed. Similar peak capacity is observed at low temperature than at high temperature with the modeled proteins. This may be helpful in removing the potential of detrimental on-column protein degradation. Second, fast gradients can be used without losing efficiency. And thirdly, a small increase in TFA may be required for sharp chromatographic peaks. Now that we have reviewed how one would go about developing a protein method, we will look at some real-world samples that were analyzed using the FLAIR Wide-Pore material. This first example is about the purity evaluation of interferon alpha-2A containing pharmaceutical products. Interferon alpha-2A has been successfully used in the treatment of several diseases such as hepatitis C. The two main oxidized and reduced forms of this 19 kilodalton protein are separated from the native form within 6 to 7 minutes, as demonstrated in the two overlays that are presented in this slide. Now, this is quite impressive, the ability to evaluate the purity of this particular therapeutic in a matter of minutes. It should be noted that the main oxidized forms are eluted before the intact form of interferon alpha-2A, It should be noted that the main oxidized forms are eluted before the intact form of interferon alpha-2A, and that the reduced forms elute shortly after the intact form. Now, back to the details of the method. The chromatographic conditions were optimized by systematically varying the gradient span and steepness, and of the column temperature and flow rate. It was found that a 7-minute long gradient at a flow rate of 0.3 milliliters per minute was appropriate to separate the related oxidized and reduced forms of interferon. Okay, filgrastim is probably one of the proteins that is produced by most big pharma companies. It is used to treat neutropenia. Here presented is a separation of filgrastim intact and its variants. The oxidized forms, which are in the red trace, are separated from the native form of filgrastim. Also, the reduced form, which is the green trace, was separated from the native form. All of these variants were separated at elevated temperature in 4 minutes. Accordingly, this fast and effective method can allow for improved throughput of purity screening. Also, this method is useful to compare biosimilar products. Because filgrastim is the first biosimilar approved in the United States, here is the current USP method for QC of filgrastim. It notes that the method uses a 4.6 by 150 millimeter C18 silica column that is operated at 65 degrees Celsius and at 1 milliliter per minute. The proposed gradient takes 25 minutes. Using the method presented in the previous slide, the FLAIR Wide-Pore column can analyze filgrastim in about 4 minutes. As such, you can reduce your analysis time by 6-fold, along with solvent consumption being reduced by a factor of 25. As can be seen with the cartoon, when a sample is able to be analyzed with both a reduction in time and solvent consumption, that is money in the bank and should put a smile on your manager's face. Other biomolecules of interest are monoclonal antibodies, or mAbs. These are an important biopharmaceutical with a wide range of clinical uses. During production, formulation, and storage, they're susceptible to chemical and enzymatic modifications. Because mAbs have therapeutic uses, they should be critically evaluated since degradation products could lead to serious health implications. Common modifications of the primary sequences are listed in the slide, such as N-glycosylation, methionine oxidation, fragmentation, lysine truncation, deamidation, and Fab amino termini variants. mAbs are subjected to a suite of analytical methods, with reversed phase liquid chromatography being one of them. In the next few slides, we will address the separation of intact and modified mAbs. There are three main approaches in reverse phase liquid chromatography analysis of mAbs. First, the complete proteolysic digestion, that is, peptide mapping or bottom-up approach. Second, the analysis of large fragments or domains, such as heavy chain, light chain, Fab or FC, FD domains. This is often called a limited proteolysis approach. And lastly, the analysis of intact mAbs. It is well known that reversed phase separation of mAbs is problematic due to their large hydrodynamic radii, the huge number of charges on these molecules, and their very low diffusion coefficients. However, to improve chromatographic performance, an optimized method for mAbs analysis will be discussed. This particular mAb is used for the treatment of a variety of cancers. In reverse phase liquid chromatography, it is known that high temperatures are needed for good separations. As such, 90 degrees Celsius was used. This produced some interesting chromatography. It is apparent that the recovery was excellent. The analysis time is under 3 minutes for both intact and reduced forms, as can be seen in both the blue and red traces. It should be further noted that the intact chromatogram plotted in the blue trace, there are 5 variants that are identifiable in a short amount of time. Furthermore, the red trace is of the reduced forms of this mAb. The reduction was performed by adding a small amount of thiotriol to the sample and then thermostating it at 40 degrees Celsius for 30 minutes. After the reduction, both the light chain and heavy chain variants are presented and are separated from the intact form. Here is another example of some heavy chain variants of IgG2 type mAb. Once again, as can be seen in the chromatogram, both light chain and heavy chain components are separated from each other. Additionally, heavy chain variants, which are seen at the end portion of the chromatogram of this mAb, are identifiable in a reduced amount of time. Once again, here's another example that shows the ability to separate several variants of the Fab fragment of the FC portion of this mAb. It's interesting that 4 different Fab variants are present in this particular method. Additionally, the separation occurs in less than 4 minutes. The digestion of this particular mAb was initiated by adding papain, then storing the sample at 37 degrees Celsius for 4 hours. This example shows the separation of IgG1 type mAb. Light chain and heavy chain and light chain SFC and FD. The SFC and FD fragments were created by papain digestion plus reduction of the antibody. Okay, let's look at the three chromatograms that are presented. The first black trace is of the intact form of this particular mAb. After reduction with papain, both light chain and heavy chain portions are presented. Further, after papain digestion and reduction, the light chain, SFC, and FD forms are present. Looking at the red trace, one could see that some of the FD variant fragments were separated. And on to our last example. Here we show the separation of Lys-C digested antibody drug conjugate, or ADC. In this case, since we have smaller fragments of the antibody, 60 degrees Celsius was sufficient to have appropriate recovery and peak widths. The analysis time was also acceptable. And of course, the separation power can be further improved by using longer columns or column coupling of columns into series. So to wrap everything up, let's review a few items that were presented in this discussion. The FLAIR Wide-Pore material is efficient in separating large therapeutic proteins. It was shown using some model proteins that separations can be accomplished at lower than usual temperatures. When working with mAbs, it is known that high temperature separations are required. Accordingly, the FLAIR Wide-Pore material is useful for these high temperature separations. A head-to-head comparison of FLAIR Wide-Pore with the best silica-based material produced comparable peak capacity. The retention is somewhat lower compared to silica-based fully porous particles and core shell materials, but can easily be adjusted by decreasing the solvent strength or by increasing the amount of TFA. We would like to thank you for your time today and would like to recognize these people listed here on this slide. For further information, please feel free to browse diamond-analytics.com. Also, you can contact our team at info@diamond-analytics.com. Now, I'll turn the time over to Laura.
Thank you, David. It's now time for the question and answer period. And as a reminder to all of you in the audience, if you would like to ask a question, you can submit it by typing it in the Q&A box, and you can find that by clicking on the red Q&A widget at the bottom of your window. So I want to ask you two questions together, David. So the first one is something you talked about at the beginning, but why diamond? And then how does the diamond compare in terms of the material and the columns made from it? How does that compare to silica?
Okay, so back to the first question regarding why diamond. At the beginning of the presentation, it was noted that the material characteristics of diamond allow it to operate in both extreme portions of the pH range. So back to that slide. So back to the slide, why diamond? So if we look at the thermal stability, diamond is able to operate at any temperature without reduction of its characteristic properties. Also, under low and high pH, comparing it to silica-based material, the material will not dissolve and or the attached ligands, for instance, the C18 ligands, will not come off of the base material. Also, the thermal conductivity of the material is large, therefore thermal gradients across the column and internal gradients can be reduced. When comparing it to silica-based materials, the pH limits are increased. As such, our operating conditions can reach from pH 1 to 13. In a particular pH study where we were looking at the low pH stability, we were using a mobile phase that contained 2.0% TFA. When we measured this in the aqueous portion, it produced a 0.7 pH value, which using a silica-based material would cause a reduction in retention over a series of column volumes because of the hydrolysis of the C18 ligands from the silica surface.
Okay. So then I'm hearing that main benefits are the ability to operate in a larger range of pH and temperature. Can you remind us what those ranges are of temperature and pH?
Sure. Yeah, so the usable pH range is from 1 to 13. And also, as shown here on this plot, it may be a little bit difficult to see. So right here we have our pH stability of 1 to 13, and then also the usable temperature stability or temperature range is up to 100 degrees Celsius.
So then how about pressure? What's the maximum pressure these columns can handle?
So here in our lab we have a UHPL system. And I've used it all the way up to its pressure limit when working with 2.1 millimeter diameter columns. Those particular columns are produced to be able to operate at those higher pressure limits. I was able to work up towards 15 to 17,000 psi. However, when working with a 4.6 millimeter ID column, the specifications on the housing material itself is around 10 to 12,000 psi.
Okay, and you just made an allusion to size. What is the maximum size at which these columns are available?
Currently, we are working on analytical type columns formats. So we offer 2.1 and 4.6 millimeter formats. However, we're open to customizable column formats. As such, if you have any questions regarding that, please send us an email at info@diamond-analytics.com, as can be seen on this slide that's on your screen at the moment.
Okay. How does the column perform under LCMS conditions, such as using ammonium formate or formic acid?
So using ammonium formate and formic acid, so if you're looking at like a buffered situation, we have limited applications using ammonium formates. However, using formic acid, which can tend to be a little bit more LCMS compatible, we've seen improved separations, but depending upon the application that you're using. And that goes back to methods development. If TFA isn't suitable for your type of application, please feel free to use formic acid in the application and developing of your method.
What is the loading capacity compared to silica-based sorbents?
So the loading capacity, we actually didn't include that information in here. So we were looking at bovine serum albumin as our model protein, and we were able to load onto a 4.6 by 50 millimeter ID column about 150 micrograms of that particular protein. We alluded to this earlier in the presentation that due to the thin shell, there is a possible reduction in sample capacity. However, as speaking with a few of our colleagues and some industrial contacts, loading capacity hasn't been an issue with our column that we've let them use.
Very good. What is the repeatability between column lots?
Repeatability between column lots, yeah, we didn't address that here in this particular presentation. However, due to our lean manufacturing type of mentality that we have here in-house, reproducibility is within 5% batch to batch and column to column.
Very good. This next question is kind of a multi-part question. What size molecule do we see, with what size molecule do we see diminished benefits? Would peptides be compatible with this column format? And what about beta amyloid?
So back to beta amyloid, I'm not familiar with that type of protein. However, as I said earlier, we can definitely look into that, and please shoot us an email regarding that, and we'll follow up to you regarding your particular application. Back to the smaller proteins. So the smallest protein that was presented was probably insulin in this presentation, as can be seen here. So insulin, its molecular weight is roughly 6 kilodalton, which is a fairly small protein. As can be seen here, using a core shell material, as you go to a smaller type of analyte, the diffusion coefficient of it is much quicker. When we're operating at 0.2 milliliters per minute and 1.0 milliliters per minute, we're roughly getting the same type of chromatography. So at the lower the lower molecular weight size of these proteins, we're seeing roughly the same chromatography, indicating that at smaller proteins that it will be okay. However, to address the peptide question, we have not yet looked at type of digests. That is in process with our methods development chemist, who's looking at different types of methods to produce good chromatograms regarding peptide analysis. However, peptide analysis, usually you use a smaller pore size particle, which would be about 120 angstroms. We do offer that type of material. However, with the larger pore size of 250 angstroms, it might not be suitable for that particular application.
Makes sense. Thank you very much, David. I think we'll wrap up now. Thanks so much for your talk and for answering all those questions.
Thank you.
I'd also like to thank all of you in the audience for participating today. And please see on your screen that there's a survey. We ask that you take that quick survey to help us, to give us feedback for future web seminars. I'd also like to thank our sponsor, Diamond Analytics, for making today's webcast possible. Today's webcast will be available for on-demand viewing through May of next year. You'll receive an email from LCGC alerting you when this webcast is available for replay. We invite you to forward that announcement to your colleagues who might have missed today's live event. We hope to see you all next time. Goodbye.